Okay, this video is for 1615 instructors. What we are looking at is the chromosome model kit to examine the different phases of meiosis. What we have here are two sets of homologous chromosomes. Our 2n is equal to 4. So we have 4 chromosomes. The different colors represent homologous pairs. So red would be a maternal chromosome and yellow would be a paternal chromosome, and same here. The different sizes represent the non-homologous pairs. So this, for example, would be chromosome pair number 1, and this would be chromosome pair number 2. These little magnetic pieces are to represent centromeres, and what we are showing here are duplicated chromosomes. Students are asked to fill out a table to describe the number of cells, number of chromosomes per cell, and number of DNA molecules present during each phase. So at G1 of interphase, what we want students to photograph, and they will be playing with these models and photographing them each phase. So what we want them to have for G1 of interphase are 4 chromosomes. They are 2 homologous pairs, and the key here is that they are unduplicated chromosomes. So these are 2 separate chromosomes. They're magnetic, so they like to be next to each other, and their photograph should look something like this. During G2, they should be able to bring back the sister chromatid because G2 of interphase happens after S phase where DNA synthesis occurred. So now we have 4 chromosomes, and they are duplicated chromosomes, so that would be 8 DNA molecules. During prophase 1, the homologous chromosomes come together and they are paired. So we still have 4 chromosomes, 8 DNA molecules because the chromosomes are duplicated. During prophase 1, as you can see, students should take a picture where the homologs are together. During metaphase 1, the homologous pairs line up at the metaphase plate. The photograph should look like this. Chromosome number, DNA molecule number is still the same, and we still have 1 cell. During anaphase 1, what students should show in their photograph is a separation of homologous pairs. This is a really good opportunity to discuss the law of independent assortment, and that they can have many different combinations of how chromosomes line up at the metaphase plate and how they are separated into daughter cells. Here we have still, in anaphase 1, it's still 1 cell. We still have 4 chromosomes and 8 DNA molecules. During telophase cytokinesis, telophase 1 and cytokinesis, what students should show you is basically something that looks like this. They could possibly move the pairs further apart from each other, and this is the point to make where the nuclear envelope reforms, and now we have 2 daughter cells, each with half the number of chromosomes. So 2 chromosomes here, 2 chromosomes here, and we now have 4 DNA molecules because the chromosomes are duplicated still at the end of meiosis 1. Prophase 2 begins with a haploid cell with 2 chromosomes. They are non-homologous and they are duplicated. So there are 2 chromosomes, 4 DNA molecules. During metaphase 2, the duplicated chromosomes, the non-homologous pairs, line up at the metaphase plate, and it looks like this. Anaphase 2 can look something like this, where the sister chromatids are pulled apart. A point to make to students is that meiosis 2 is very similar to mitosis in that sister chromatids are pulled apart and you end with unduplicated chromosomes. So during anaphase 2, sister chromatids are pulled apart. We now went from 2 chromosomes to 4 chromosomes. there are 4 DNA molecules, 1 cell. Telophase 2 and cytokinesis is where we now form 2 separate cells with different chromosomes. So we have the nuclear envelope reforms, and we have the result is 4 haploid cells. So we can have students show their models like so. And again, this is a good time to talk about the law of independent assortment. Sorry, the magnets just keep pulling the models together, but telophase 2 and cytokinesis results in 4 daughter cells that are genetically distinct.